Hi, Mr. S here. Welcome to my video on the kidney. In this video we're going to look at kidney structure and the function of the kidney, which is to filter the blood of toxins and to control fluid levels of the blood. Okay, so the kidney is all about the plumbing. We have the blood system taking blood into the kidneys, and we've obviously got veins going away from the kidneys. We've also got the connection, the interface between the blood system and the urinary system. As the urinary system on this diagram is yellow, and we have the ureter, which is taking urine from the kidneys to the bladder where it's stored, and we have the urethra, which is taking urine out of the body. How I remember this is ureter goes to the bladder and urethra goes through and outside to the outside wall. At this point we're going to have a look inside the kidneys, so if I just take a cross section there, we can see a little closer about the plumbing and the connections between the two systems. So I've zoomed in here and we can see we have the left kidney and we have the right kidney. Same rules apply to labelling these as any other organ in the body. Imagine you are the patient and the left will be this side and the right will be this side. We can see here we've got the blood moving into the kidney through the renal artery here. At this point the blood will then break off into smaller capillaries, smaller networks, and we'll have a look at those in a bit more detail in a bit. And then we'll have the blood moving back out through the renal vein. We have the ureter, which we just saw in the previous slide, taking the urine away from the kidneys. We've also got this structure here, which is called the pelvis, not to be confused with the pelvis, the bone pelvis, but the origin of the word's quite similar. It comes from the Latin to mean basin, so it's a collecting area for urine. Again, we're going to see how this works in a little bit. The kidneys are labelled with the outside of the kidney being the cortex and the middle being the medulla. Again, cortex we remember from the brain, we also remember from the adrenal gland. Cortex means bark and it's the outside bit of the organ, and the medulla is the middle bit of the organ. So here I've zoomed in. Obviously this is an oversimplification and there are millions of these nephrons, but here's a nephron in its place in the kidney. You can see the top half of the nephron here is found in the cortex, and the bottom half of the nephron is found in the medulla. So often questions come around here, what part of the nephron you find in the cortex. So with this we've got the glomerulus, the convoluted tubules, and in the medulla we have the loop of Henle. Now if we look at the structure of a nephron, there's a lot going on here. There are a lot of words. Over time they will stick. We have blood coming in to the nephron through this arteriole, and this is called the afferent arteriole. The blood then will move through a capillary network here, and this is called the glomerulus. A tricky mouthful to say, but glomerulus. I think that comes from the Latin to mean a ball of twine, which is quite apt really if we zoom in. So there's the glomerulus. Surrounding the glomerulus is something called the Bowman's capsule. So in this diagram we can see the blood system here being in the red, and in yellow we have the urinary system. So at this point this is the interface between the blood system and the urinary system. This is where exchange will take place. So blood leaving the glomerulus will be through the efferent arteriole. We have a zoom out again. So the efferent arteriole will be taking blood away from the glomerulus. We can then see the blood branching off through a whole load of arterioles moving around here, and forming and then branching off into what will then become the renal vein. A point as we go through this is to remember that surrounding the urinary system will be branches of the blood system. Okay? Moving on to the urinary side of things now, we've got the Bowman's capsule with the filtrate in, and that will move through the first bit of tubule here, which we call the proximal convoluted tubule. Convoluted just means winding, and proximal to mean close to. So the closest part of this tubule to the Bowman's capsule is called the proximal convoluted tubule. The proximal convoluted tubule then will move through this, which is the loop of Henle. So the whole thing here is called the loop of Henle, and we will discuss the loop of Henle and what goes on the loop of Henle a little bit later. The urinary system then moves through the distal convoluted tubule, so distal to mean distant, so far away from the Bowman's capsule, and then that will drain into this, which is called the collecting duct. Functions of all these areas we will look at now.
So we've now zoomed into the glomerulus, and I've taken a cross section here, and we can see the Bowman's capsule surrounding the glomerulus. We can also see here the blood, as we've just in the previous image looked at. We've got the blood moving around this capillary bed and coming in via the afferent and then out via the efferent arterial. Notice a very key point here: the efferent arterial is narrower. The lumen of the efferent arterial is far narrower than that of the afferent arterial. The reason for this is it's going to increase the pressure within the capillaries of the glomerulus. Okay, so we've got some more keywords now. Some you don't need to know. For example, the fenestrae is not something you need to know. However, I think it helps understanding if we can talk about it. We've got podocytes, which I'm highlighting now. These you do need to know, and we'll cover those in a little bit. If you can't get the concept of what they are yet, don't worry. And we've also got something between the capillary bed, the glomerulus, and the podocytes, which is the basement membrane. Now, this basement membrane is made of collagen, and again, the structure and function of this is really, really important and key to the functioning of the glomerulus and Bowman's capsule, and we will go a bit more detailed as we move on. If I zoom in a little bit closer, we can see how the high pressure in the capillary is going to have a function here, which is to push the liquid part of the blood out through the fenestrae, and the fenestrae are simply gaps in the capillary wall, so they're almost pores. So the liquid part of the blood will be moving out through these fenestrae and out across the basement membrane. Now, so the basement membrane was collagen. It was a fibrous network of collagen, and the function of this is simply to prevent the blood cells leaving the blood. So just the liquid content will be moving out. We can also see the liquid content moving out across these podocytes and into the Bowman's capsule. At this point, we'll refer to it as filtrate, and here on in, we'll be talking about this being the filtrate until, of course, it becomes urine, the finished product. So I've zoomed in on a single podocyte, which is simply a cell of the lining of the Bowman's capsule, and it grips around the capillary, and what it will do is prevent, if you can see here, the plasma will be moving out through these gaps in the podocyte, and what this does is it acts as a second barrier beyond the basement membrane to prevent the flow of cells from the blood into the filtrate. We can see here the basement membrane, so that's the collagen fibers between the podocytes and the capillary wall. Here we have the overall structure. We've got the arrows representing the movement of the plasma out of the blood into the Bowman's capsule. Moving on a little bit further now, we've got the filtrate moving from the Bowman's capsule into the proximal convoluted tubule. And at this point, everything has left the blood in terms of hormones, amino acids, glucose, obviously the urea, and it's not selective, so basically everything that is in the liquid part of the blood has moved out. The only thing that hasn't really is going to be the blood cells, and obviously some plasma as well, because clearly blood wouldn't be able to flow without it being liquid. So not everything passes out, but the majority of it does. So within this, as I said, will be amino acid, glucose, vitamins, hormones, and at this point in the proximal convoluted tubule, we're going to be reabsorbing back into the blood. So we can see here the proximal convoluted tubule having close connections with the blood vessels, and we're going to be seeing the good stuff, so the amino acids, the hormones, the vitamins being reabsorbed back into the blood. So around 85% of the sodium, chloride, the salts and water is also reabsorbed as well back into the blood at this point. If I zoom out, what I've got here is I've got a representation of the walls of the proximal convoluted tubule. So I've just zoomed in. If you can see here, we've got a little red box here. So I'm literally representing the wall of the convoluted tubule here. The inside of this is obviously going to be the filtrate, so we've got movement of the filtrate past these cells. We've also got a blood vessel surrounding the convoluted tubule. In the middle, we've got a gap, so that will be tissue fluid. So between the walls of the proximal convoluted tubule and the capillary, we've got tissue fluid. We're going to get at this point active transport from the filtrate. So the good stuff I mentioned, the glucose, the amino acids, will be actively transported across the cells of the lining of the convoluted tubule and into the surrounding tissue fluid. Make note obviously of the mitochondria making links to active transport here. The cells of the proximal convoluted tubule have loads of mitochondria for the active transport to take place, and they've also got microvilli on which obviously increase the surface area and increase the rate of uptake of amino acids and glucose. As we get across here into the tissue fluid, we've got the movement of the glucose, the amino acids, back into the blood, and this will be via diffusion. Obviously we have a concentration gradient here, so loads here, not much in the blood obviously because it's just come from having got rid of all of its glucose and amino acids, and it will be passing by with very low levels in the blood. So we're going to get diffusion from a high concentration to a low concentration in the blood. Also nice to mention the fact that we've got movement of blood away, so we're maintaining a concentration gradient. We've now got the filtrate moving through the loop of Henle. Now this I'll pick up in a bit. It's going to be about absorption of water, and I will pick that up in a moment. The filtrate will now move through the distal convoluted tubules, and at this point and the collecting duct we have fine control of water levels, and this will be controlled by the hormone ADH, antidiuretic hormone. Again, I'll pick that up in a bit. At this point in the collecting duct we have our levels balanced. We've got within the urine we've got urea, and we're going to have the urine moving through the collecting ducts into the pelvis and then into the ureter and off to the bladder. So to summarise at this point, we've got the renal vein, or branch of the renal vein, taking blood away. That will be high in glucose levels, amino acids that have been reabsorbed back into the blood in the proximal convoluted tubule. We've also got water and salts that have been balanced through the loop of Henle, and we've got the urine being taken away in the collecting duct. So we'll go back to the loop of Henle, but here we are, and we're going to look at something called the countercurrent multiplier. This is obviously the mechanism for reabsorption of water back into the blood. Some things to note before we move on. This image I have is obviously missing the distal convoluted tubule, so forgive that, but do not forget that we have the distal convoluted tubule here between the loop of Henle and the collecting duct. We have a descending limb of the loop of Henle. We have an ascending limb of the loop of Henle. So downwards, descending, upwards, ascending. Two things to note about these. One, the ascending limb of the loop of Henle is permeable to sodium and chloride ions, not to water. So the walls of the ascending limb are impermeable to water. The descending limb is permeable to water. One other thing I should note is that surrounding the loop of Henle, if you remember, we would have the capillaries. Now, I have taken those off from this diagram, but please be aware that surrounding the loop of Henle will be the capillaries. Another key word we need to know or talk about is osmolarity. Now, osmolarity refers to the measurement of how much of a solute is dissolved in a solution. So it's a bit like water potential that we did in year 12, where 0 is pure water. There's nothing dissolved in it, so it has a value, an osmolarity value of 0. As we increase the solute, so we're dissolving more solutes in this solution, we're making it more concentrated, the osmolarity increases. The higher the number, the greater the concentration. Our story starts with the filtrate being isotonic with the surrounding tissue fluid. This means that it has the same osmolarity, so it will be the same concentration as the surrounding tissue fluid. I'm now going to skip on to the ascending limb of the loop of Henle. We'll tie the two up in the middle, but for now we're looking at this, the ascending limb of the loop of Henle, and at this point we've got active transport of sodium ions and chloride ions out of the filtrate into the surrounding tissue fluid. Now you can probably work out what this is going to do to the surrounding osmolarity of the tissue fluid and the filtrate. So we can see sodium ions and chloride ions in the surrounding tissue fluid, the concentration increasing, osmolarity increasing. We've got 400 now, and as we said before, the osmolarity of the filtrate will have decreased because it's losing solutes to the outside surrounding tissue fluid. How does this happen? Well, if we can make links here, we'll be talking about intrinsic proteins in the plasma membranes, and we're moving sodium ions through sodium pumps and chloride ions through chloride pumps, and it will be an active process. As I said, the osmolarity of the tissue fluid, I call it interstitial fluid, but you can call it tissue fluid absolutely fine, it's not a problem, and this will increase. So we've now got a osmolarity of 400 in the tissue fluid of the medulla, if you remember, the medulla being the part of the kidney that has the loop of Henle's in. Now the consequence of this increase in osmolarity is that the filtrate moving through the descending loop of Henle, we've already established being osmolarity of 300. These are arbitrary units I'm giving you here, but they represent what's going on quite nicely. As we have a lower osmolarity in the filtrate to the surrounding tissue fluid, we're going to get movement through osmosis out of the filtrate into the surrounding tissue fluid, because there is a gradient, a concentration gradient. Now, another point to make is that I'll be concentrating on the descending loop of Henle, but of course the collecting duct is surrounded by this tissue fluid as well, so there will be a concentration gradient here as well. So you might get some movement of water out from the collecting duct, but for now we're just talking about the descending loop of Henle. Now if you remember, the ascending limb, the loop of Henle, is impermeable to water. So the more dilute filtrate that we have now will just move on up through and will not lose any water. If you remember, over this side we've got movement of water because of the concentration gradient. There is a concentration gradient here, but what stops the water moving out is the fact that the walls are impermeable to water. Okay, so we have an increase in osmolarity of the filtrate here, and this will be moving round and down through the descending loop of Henle. You can see here just moving down through the loop of Henle. This will replace the dilute filtrate. So we've got the more concentrated filtrate moving up through here, replacing the dilute filtrate. And at this point, we've got the movement of more sodium chloride ions out of the filtrate. So we're moving more sodium chloride ions out of the filtrate into the surrounding tissue fluid or interstitial fluid of the medulla. And this will further increase the concentration or the osmolarity of the surrounding tissue fluid. We will now see, therefore, a concentration still for the higher osmolarity filtrate. So this filtrate that's increased in concentration or has a higher osmolarity will still lose water because of the concentration gradient. It's also important to note here what happens to the water. So obviously we've got an increase in water in the interstitial fluid here. But if you remember, I took away the blood vessels, but we have blood vessels passing past this water, so water will move from the interstitial fluid into the blood vessels, and then obviously back around our body. The cycle repeats, so more salt will leave, will increase the osmolarity of the surrounding tissue fluid, and we're going to get a movement of water from a higher concentration or higher osmolarity filtrate over and over again, and it just depends on the length of loop of Henle. Clearly this is a process designed to increase the amount of water conserved from urine. Without this process we'd lose far too much water in our urine. So this slide shows a longer loop of Henle in desert animals. Obviously the longer loop of Henle will conserve more water, so there's more distance for the countercurrent multiplier to happen over, therefore more water will be conserved. Normal or mesic animals will have a shorter loop of Henle and therefore not so much water conservation. That is, after all, not that necessary. Obviously very necessary for desert animals to conserve water. Questions often come out about length of loop of Henle and why or how that increases the amount of water that is conserved. So having obviously got the balance right at the water levels of the loop of Henle, we're now moving through the distal convoluted tubule. If you remember up here, the distal convoluted tubule and into the collecting duct. So the collecting duct will now take away the end product urine away to the pelvis, and then obviously to the ureter, and then to the bladder. So as I mentioned earlier, the distal convoluted tubule and the collecting duct are affected by ADH, just a fine control of water balance, and we'll have a look at how that can be now. Now I've just zoomed into a part of the collecting duct, so we have represented by the blue on the outside of the collecting duct being the interstitial fluid or the tissue fluid, and we've got the collecting duct cells here. So there's a collecting duct cell, so that just represents the black line there, and we have the other side of the vessel here, which is represented by the black line here. The yellow in the middle is obviously going to be our urine. So with that said, let's have a look at how ADH can affect the permeability of these cells to water. Now you might remember from GCSE, ADH, antidiuretic hormone, is produced when you are dehydrated. So the idea is that the hormone will be produced to increase reabsorption of water from our urine. So we're conserving more water from our urine. And this is obviously controlled by the hormone ADH. So we have the ADH here, and you'll see ADH receptors on the cell surface of the collecting duct cells. That will tell you, if you're going back to hormones, and you're probably already thinking this through, that it is a non-steroid hormone. It cannot pass through the plasma membrane, so it will have receptors on the surface of the plasma membrane, and it will act through, you guessed it, our second messengers. Here we can see our favourite second messenger, CAMP, and we've got ADH having bound, and we've got the release of CAMP, which will cause a cascade of events inside the cell. And I think these are pretty cool. So we've got a vesicle here. So this is a vesicle, and the CAMP will cause the vesicle, or the CAMP causes the cytoskeleton to move the vesicle. to the plasma membrane. And within the vesicles, these are special vesicles. Within the membrane of the vesicle, we've got a protein called aquaporin. So what aquaporin is an intrinsic protein within the membrane, which is a channel protein, and it allows for the bulk movement of water across a membrane. Aquaporin. The vesicles will bind with the plasma membrane of the cell, and through exocytosis, the content of the vesicles are irrelevant. It's the fact they bind and they go through exocytosis, and they will become part of the plasma membrane. So that vesicle, having become part of the plasma membrane, will obviously incorporate its aquaporin into the plasma membrane, and that causes or allows a pathway for water to move into and through the cell, thus retaining water from the urine and conserving water. So there we go, that's that. If you've got any questions, come and find me. You might want to look back over your hormones notes. You might also want to look back over your plasma membrane notes from year 12. Hope it's alright, and I'll see you again soon.